viewers, welcome to CEC Gurukul Lectures. I am Dr. Sanawar Soham from Department of Botany, Kalindi College, University of Delhi, and I will be discussing biometrics, numerical taxonomy and cladistics in this part of my lecture. Now, to begin with, we shall understand the meaning of term biometrics. Biometrics is derived from the Greek words which means bio, we all know, life, and metrics means to measure. Now, biometrics is the term that encompasses the application of modern statistical methods to the measurements of biological objects. Biometric identification is a pattern recognition based classification system that recognizes an individual by determining its authenticity using a specific physiological or behavioral characteristic. The emerging biometric and bioinformatic approaches to plant sciences are critical for better understanding of how taxonomy relates to other fields of botany like ecology, physiology, genotype, evolutionary and phylogenetic history. From microscopic pollen grains and charcoal particles to macroscopic leaves and whole root systems, the methods presented include automated classification and identification, geometric morphometrics, skeleton networks, as well as tests of the limits of human assessment. All demonstrate a clear need of these computational and morphometric approaches in order to increase the consistency, objectivity, and throughput of plant taxonomical studies. There are many characteristics that are uniquely identifiable at a species level, and these can be easily measured. Biometrics can be strengthened by adding reference images of new plant species to the database or by adding mere reference images of existing species. In human recognition, if we talk of fingerprints, face, iris, speech and hand geometry are the most commonly used biometrics, and a number of commercially available biometric systems for human recognition are available in market today. But in contrast, for plants, even though they have many characteristics that are uniquely identifiable at specific level, there is not such a biometric system for plant identification. So this is the study area of plant taxonomy which has gained importance in recent times, and we are working towards it. Biometric authentication systems generally use a pattern recognition approach that verifies or identifies an organism's authenticity of its uniquely identifiable physiological or behavioral characteristic, which is called biometric. Verification systems either reject or accept the submitted claim of identity by comparing it with the pre-stored biometrics of the same individual, while identification systems recognize an individual by searching the matching queried biometric in the entire database. Now, we will move on to the next part of our lecture, which is numerical taxonomy. Today, taxonomy is different from what it was a generation ago. The use of computers by taxonomists has established an interesting modern trend called numerical taxonomy or taximetrics. Mathematical and statistical evaluation of taxonomic information and computation of this data has provided taxonomists with new approaches to understand classification. The use of computers in numerical taxonomy has made it possible to compare a large number of characters from many organisms with relative ease. And after comparison of the organisms, they are grouped according to the overall similarity or dissimilarity wherever necessary, and it is presented graphically. M. Addison was the one who proposed that a classification should use a vast range of characters covering all aspects of plants, and in construction of a classification, all characters must be given equal importance. This idea forms the basis of modern numerical taxonomy, which is also called Neo-Addisonian taxonomy. So what is numerical taxonomy? It is an organized method of evaluating data with computers in an objective and repeatable manner, enabling comparison of many characters from many populations of plants. The book Numerical Taxonomy by Sneath and Sokal provides valuable information relating to the aims, principles, methods, and results of numerical taxonomy. The proper selection of characters is the critical point in the application of numerical taxonomy. So once we are moving towards the understanding of how to go about the numerical taxonomy, we need to understand some important attributes and aspects, one of them being the characters. So we have to choose some characters which are important in understanding or in grouping of organisms. So certain characters are clearly disqualifiable for numerical taxonomy, and these are listed by Sokal and Sneath as inadmissible characters. According to them, it is undesirable to use the following characters. For example, the attributes which are not a reflection of the genotype of the organisms themselves. Secondly, any property which is a logical consequence of another, either partly or wholly. The third one being the characters which do not vary within the entire sample of organisms. They are also undesirable for numerical taxonomic purposes. They also suggested that there are some characters which are admissible, and they are that it should be inherent in the organisms, the characteristics which are inherent in the organisms and should neither be susceptible to environmental changes nor are affected by the experimental uncertainties, are the ones which should be chosen for numerical taxonomic purpose. Secondly, the character must be of some diagnostic value. It should have some demarcating or diagnostic feature which helps us in grouping or which helps in basically classifying the organisms. The third one being it cannot be subdivided, and the fourth character is that the character is selected must show variation among the taxa under comparison. So we'll now discuss the principles of numerical taxonomy. The first one being the greater the content of information in the taxa of a classification is, the more characters on which it is based, the better a given classification will be. Obviously, we very well understand if we have a rigorous data and if we have many characteristics which can be compared in various taxa, obviously our classification will be more refined. The second principle of numerical taxonomy states that every character is of equal weight in creating the natural taxa. So, the weightage, that is importance to some characters and we actually giving no less importance to some other features, are not basically the principles of numerical taxonomy. In numerical taxonomy, we basically give equal weightage to each and every character being studied. Then the third principle of numerical taxonomy is that the overall similarity between any two entities is a function of the individual similarities in each of many characters for which they are being compared. So even if there are some features which are common, the overall similarity is found out between the taxa to rule out the basically closeness of various taxon. The fourth principle is distinct taxa can be recognized because correlations of characters differ in the groups of organisms under study. The fifth important principle of numerical taxonomy is that phylogenetic inferences can be made from taxonomic structure of a group and from character correlation given certain assumptions about the evolutionary pathways and mechanism. And the sixth one being that today taxonomy is viewed and recognized, we know, as an empirical science. So obviously this is the basis of numerical taxonomy, where we give more weightage or we give more emphasis on the observations, the comparisons, rather than just the history or the basically the logic. The seventh principle of numerical taxonomy is a classification is based on phonetic similarity. We all have very well understood the concept of phonetics, the phonetic classification. So I will now move on to the next part, that is numerical taxonomy and how we basically create or how we go about classifying an organism based on numerical taxonomy. So numerical taxonomy is a new method of organizing data that may prove helpful in better understanding of relationships. The number of characters studied in numerical taxonomy is usually about 50 to 100 from approximately the same or greater number of organisms. The greater the number of characters, as I already told you, the more valid the classification will be. Numerical taxonomy is an operational science. The procedure is divided into a number of repeatable steps. We will now move on to the steps of numerical taxonomy. The first step is to decide what kind of units we want to study. In numerical taxonomy, the basic unit is called the operational taxonomic unit, or OTU. OTU is basically the term given to the lowest taxon being studied in a particular investigation. So what is an OTU? The OTU then? OTU can be an individual plant for the taxonomist if a single population of a plant is studied, or if we are studying a single population of plant to find out the range of variations in its character. OTU, if we treat an entire population of plants as an OTU we are studying, then a single species can be represented by different populations existing in nature. OTU may be different species when we are basically evaluating a genus. So OTU varies. It can be a population, it can be a species, it can be a genus, depending upon what are we studying. Therefore, in numerical taxonomy, the OTU varies with the material being studied, and this helps the taxonomist in making an objective study. Now, after the selection of OTUs is done, it is necessary to select characters by which they are to be classified. The characters, as I already told you, there are some admissible ones, some non-admissible ones, and we have to find out the diagnostic characteristics which are to be compared. So the characters which vary greatly amongst the OTUs are clearly more useful in numerical taxonomy. And we know that as many characters as possible can be used. This is known as character selection. Now generally 60 characters should be considered a minimum, whereas 80 to 100 or more characters are needed to produce a fairly stable and reliable classification. The selected characters have then to be coded or given some symbol or mark. There are two methods of coding the taxonomic information. The data on the characters of OTUs may be suitably coded. The selected characters have then to be coded or given some symbol or mark so that the data can be computed. And how we do that? For this purpose, a general listing of all the characters is very helpful. It is important to treat all characters as having equal importance, a priori weighing, which means equal weighing, as this is one of the 7 principles of numerical taxonomy which I already discussed. Now for coding of characters, as I told you in the previous slide, there are 2 ways or methods of coding taxonomic information for the purpose of numerical taxonomy. They are binary coding or two-state coding, and the second one being multi-state coding. Now let's discuss binary coding or two-state coding. This is the simplest form of coding adopted in numerical taxonomy, where the characters are divided into plus or minus. They are given these signs or designated 1 or 0. The positive characters are recorded as plus sign or 1 and the negative characters as minus or 0. It is possible to use this method of coding for all characters studied. In case a particular character is not present in OTU being examined, then which symbol we give? We give NC. This indicates that there is no comparison for that character. However, by using this method of coding, we tend to increase our work. Why? Because there are large number of variations in plants and it is very difficult to basically code them into binary method like either 1 or 0, and very often a single character such as color of the flower can be represented in a wide range. So we can have white, pink, red, yellow and other flowers and roses. If we have to use this data in a binary coding, then we'll have to use each color as a character and it would be coded as this color present or absent, then pink color present or absent, red color present or absent, so on and so forth. So for that purpose, we use multi-state coding. In this, single character can be coded in a number of states, each being represented by a numerical symbol or code. For example, we give them 1, 2, 3 numbering depending upon the range of variation. If we again look at the color of the rose flower, we can give different codes to different colors instead of just as in binary we were giving 0 or 1. In multi-state, we can give designate a particular color to a particular number. For example, white is given 1, pink 2, etc. etc. So multi-state coding is also useful for quantitative characters such as plant height, length of the leaf, leaf breadth and other characters which involve measurements and have variety of range or a wide range. A code is prepared for the range of variation and appropriate symbols are then allotted to each unit in the range. Once we are through with coding of the characters, we then move on to the preparation of data matrix. So the data obtained by scoring the characters in the OTUs are then presented in the form of a table as a data matrix, giving the OTUs on one side of the table and the codes for different characters against each OTU. Thus, if say we have studied 25 OTUs and we have scored or we have studied 100 characters for each, then the data matrix will contain as much as 2500 units of information. Thus, we need computer for such numerical taxonomic purposes. The next step in numerical taxonomy is entirely dependent on the data matrix. The information is then presented as TN table or data matrix consisting of OTU scored for n characters. Now, this slide shows the coded data in the form of TNN table, where you can see on the left side there are 1 to 12 the numbers which are basically the characters, and on this side of the table you can see A, B, C, D, the 4 taxa. This is a hypothetical example where 4 taxa or OTUs say are used, and we have tried to find out and we have coded the table and we have seen the presence or absence of a particular attribute in these taxon. So moving on to then we have to calculate the overall similarity. See, once this table is formed, next step is to find out the similarity of characters in different OTUs. So how do we do that? We have to measure the similarity. The similarity coefficient is calculated. So overall similarity is calculated by comparing each OTU with every other and is usually expressed as percentage. 100% S for identical and 0% for no resemblance. A similarity table or matrix is then constructed by tabulating the S coefficient, that is similarity coefficients for each one of the OTUs. So what did we do? We tabulated first the characters and OTUs studied, and then we basically form a matrix of all the similar taxon based on the similarity coefficient. So this slide shows a similarity table or matrix where you can see that we have placed taxa on both the sides, the x and y axis, and the matrix shows that the taxon A has 100% similarity with the taxon A itself, right? And it has 80 to 90% similarity. Taxon A has 80 to 90% similarity with the taxon B. So we basically designate different symbols or different basically solid pictures we designate in the matrix once we have just made up, or we have basically placed the matrix based on the percentage similarity from say 10% to 50 or 55%. Then the next step is that we bring in together all the taxa which have similar percentage, or we basically are clustering them. We cluster them together based on the overall similarity coefficient. Now the most common way used or the method for determining similarity or resemblance is to calculate the simple matching coefficient. How it is done? This computation was first used by Sokal and Michener in 1958, and it is based on a very simple mathematical formula. We calculate simple matching coefficient with this formula where we divide the total number of matches, that is the similarity, and we divide it with the summation of the total number of matches and mismatches. And then it is better if we use or calculate similarity as a percentage. We can use this with the simple formula in the form of percentage. Now the data on percentage similarities of all the OTUs against all other OTUs can be calculated in this manner. Thus, when one OTU is compared with itself, the similarity will always be 100%. On the other hand, when no single character is common between the two OTUs, the similarity will be 0%. Therefore, by comparing each OTU with all other OTUs, it is possible to obtain large amount of information on the similarities as well as dissimilarities among the units which we investigated. Now, in the second part of the lecture, I will be starting with cluster analysis. Thank you.